In order to try and repair the brain in conditions like Huntington's and Parkinson's, you need to have a set of neurons, a set of nerve cells, which will replace those which are lost as part of the core pathology. In Parkinson's, those are nigral dopamine cells. In the Huntington's disease, whilst there's a lot of cells that are lost, the striatal DART32 positive projection neurons are the ones which are preferentially lost. In both cases, there have been clinical trials designed to try and replace those cells using fetal equivalent. So the idea is that the fetal material, you dissect out the developing striatum or the developing dopamine cells from the midbrain in Parkinson's and Huntington's disease, and you transplant them into the patient. In Parkinson's, those trials have been going on for about 20-25 years with very mixed results, probably due to a whole series of reasons to do with patient selection, how the tissue is prepared, how it's implanted, how it's supported after it's grafted, and trial design. What we do know using fetal dopamine cells in patients with Parkinson's is when it works very well, but it doesn't work in all cases. In Huntington's disease, the transplantation of the human fetal striatum into people with Huntington's disease has produced less clear-cut results. In some patients, and this has been going on now for the last 15-20 years, in some patients there clearly is an improvement. There's a patient I see here who's had a very dramatic improvement following his transplant, but again the results are somewhat inconsistent, and there are less patients who've had the transplants compared to Parkinson's to know exactly whether this is a viable therapy for the future. In both cases, I would say that there is enough evidence to suggest it's worth pursuing. The use of human fetal tissue for grafting patients will never be a viable option because of logistical and ethical problems related to it. The advent of stem cell derived equivalent of those human fetal tissues, so the DART32 projection neurons for Huntington's disease, would be very helpful because then we'd have a ready supply of cells that we could then use in patients as and when required.